Anatomy of flowering plants. You can very easily see the structural similarities and variations in the external morphology of the larger living organisms, both plants and animals. Similarly, if we were to study the internal structure, we can also find several similarities as well as differences. This chapter introduces you to the internal structure and functional organization of higher plants. Study of internal structure of plants is called anatomy. Plants have cell as basic unit. Cells are organized into tissues and in turn the tissues are organized into organs. Different organs in the plants show differences in their internal structure. Within angiosperms, the monocots and dicots are also seen to be anatomically different. Internal structures also show adaptations to diverse environments. The tissues. A tissue is a group of cells having a common origin and usually performing a common function. A plant is made up of different kinds of tissues. Tissues are classified into two main groups, namely meristematic and permanent tissues, based on whether the cells being formed and capable of divide or not. Meristematic tissues. Growth in plant is largely restricted to specialized regions of active cell division called meristems. In Greek, meristome means to divide. Plants have different kinds of meristems. The meristems which occur at tips of the root and shoot and produce primary tissues are called apical meristems. Root apical meristem occupy the tip of the root, while the shoot apical meristem occupy the distantmost regions of the stem axis. During the formation of leaves and elongation of stem, some cells left behind from the shoot apical meristem constitute the axillary bud. Such buds are present in the axils of leaves and are capable of forming a branch or a flower. The meristem which occurs between mature tissues is known as Intercalary Meristem. They occur in grasses and regenerate parts removed by the grazing herbivores. Both apical meristems and intercalary meristems are primary meristem because they appear early in life of a plant and contribute to the formation of the primary plant body. The meristems that occur in the mature regions of the roots and shoots of the many plants, particularly those that produce woody axis and appear later in than primary meristem, is called secondary or lateral meristem. They are cylindrical meristems, fascicular vascular cambium, interfascicular cambium, and cork cambium are examples of lateral meristems. These are responsible for producing the secondary tissues. Following divisions of cells in both primary as well as secondary meristems, the newly formed cells become structurally and functionally specialized and lose the ability to divide. Such cells are termed as permanent or mature cells and constitute the permanent tissues. During the formation of permanent plant body, specific regions of the apical meristem produce dermal tissues, ground tissues, and vascular tissues. Figure 6.1 Apical meristem. A. Root. B. Shoot. Permanent tissues. The cells of the permanent tissues do not generally divide further. Permanent tissues having all cells similar in structure and function are called simple tissues. Permanent tissues having many different types of cells are called complex tissues. Simple tissues. A simple tissue is made of only one type of cells. The various simple tissues in plants are parenchyma, collenchyma, and sclerenchyma. Parenchyma forms the major component within organs. The cells of the parenchyma are generally isodiametric. They may be spiral, oval, round, polygonal, or elongated in shape. Their walls are thin and made up of cellulose. They may either be closely packed or have small intercellular spaces. The parenchyma perform various functions like photosynthesis, storage, secretion. The collenchyma occurs in layers below the epidermis in most of the dicotyledonous plants. It is found either as homogeneous layer or in patches. It consists of cells which are much thickened at the corners due to deposition of cellulose, semicellulose, and pectin. Collenchymatous cells may be oval, spherical, or polygonal and often contain chloroplast. These cells assimilate food when they contain chloroplast. Intercellular spaces are absent. They provide mechanical support to growing parts of the plant such as young stem and petiole of a leaf. Sclerenchyma consists of long narrow cells with thick and lignified cell walls having a few or numerous pits. They are usually dead and without protoplast. On the basis of variation in form, structure, origin, and development, Sclerenchyma may be either fibers or scleroids. The fibers are thick-walled, elongated, and pointed cells, generally occurring in groups in various parts of the plant. The scleroids are spherical, oval, or cylindrical, highly thickened dead cells with very narrow cavities, that is lumen. These are commonly found in the fruit walls of nuts, pulp of fruits like guava, pear, and sapota, seed coats of legumes, and leaves of tea. Sclerenchyma provides mechanical support to organs. Complex tissues. The complex tissues are made of more than one type of cells, and these work together as a unit. Xylem and phloem constitute the complex tissues in plants. Xylem functions as a conducting tissue for water and mineral from roots to the stem and leaves. It also provides mechanical strength to the plant parts. It is composed of 4 different kinds of elements, namely tracheids, vessels, xylem fibers, and xylem parenchyma. Gymnosperms lack vessels in their xylem. Tracheids are elongated tube-like cells with thick and lignified walls and tapering ends. These cells are dead and are without protoplasm. The inner layers of the cell walls have thickenings which vary in form. In flowering plants, tracheids and vessels are the main water transporting elements. Vessel is a long cylindrical tube-like structure made of many cells called vessel members, each with lignified walls and a large central cavity. The vessel cells are also devoid of protoplasm. Vessel membranes are interconnected through perforations in their common walls. The presence of vessels is a characteristic feature of angiosperms. Xylem fibers have highly thickened walls and obliterated central lumens. They may either be septate or aseptate. Xylem parenchyma cells are living and thin-walled, and their cell walls are made up of cellulose. They store food material in the form of starch or fat and other substances like tannins. The radial conduction of water takes place by the ray parenchymatous cells. Figure 6.2 Simple tissues. A. Parenchyma. B. Collenchyma. C. Sclerenchyma. A fiber, a scleroid. Primary xylem is of two types, protoxylem and metaxylem. The first formed primary xylem elements are called protoxylem, and the later formed primary xylem is called metaxylem. In stems, the protoxylem lies towards the center or pith, and the metaxylem lies towards the periphery of the organ. This type of primary xylem is called endarch. In roots, the protoxylem lies towards periphery and the metaxylem lies towards the center. Such arrangement of primary xylem is called exarch. Phloem transports food materials, usually from leaves to other parts of the plant. Phloem in angiosperms is composed of sieve tube elements, companion cells, phloem parenchyma, and phloem fibers. Gymnosperms have albuminous cells and sieve cells. They lack sieve tubes and companion cells. Sieve tube elements are also long tube-like structures arranged longitudinally and are associated with companion cells. Their end walls are perforated in a sieve-like manner to form a sieve plate. A mature sieve element possesses a peripheral cytoplasm and a large vacuole but lacks a nucleus. The functions of sieve tubes are controlled by the nucleus of companion cells. The companion cells are specialized parenchymatous cells which are closely associated with sieve tube elements. The sieve tube elements and companion cells are connected by pit field presents between their common longitudinal walls. The companion cells help in maintaining the pressure gradient in the sieve tubes. Phloem parenchyma is made up of elongated, tapering, cylindrical cells which have dense cytoplasm and nucleus. The cell wall is composed of cellulose and has pits through which plasmodesmatal connections exist between the cells. The phloem parenchyma stores food material and other substances like resins, latex, and mucilage. Phloem parenchyma is absent in most of the monocotyledons. Phloem fibers or bast fibers are made up of sclerenchymatous cells. These are generally absent in the primary phloem but are found in the secondary phloem. These are much elongated, unbranched, and have pointed needle-like apices. The cell wall of phloem fibers is quite thick. At maturity, these fibers lose their protoplasm and become dead. Phloem fibers of jute, flax, and hemp are used commercially. The first formed primary phloem consists of narrow sieve tubes and is referred to as protophloem, and the later formed phloem has bigger sieve tubes and is referred to as metaphloem. Figure 6.3 A. Xylem. B. Phloem.